So as you can see, we're running a bit behind, but I want to assure you that there is the poster session, which follows this, is actually a 2-hour poster session, and we're going to try to get the coffee break, which follows this, extended. So please feel free to stay. If you have to leave because you're presenting a poster, of course, we understand, but we hope that you can stay for the last talk. And as they're setting up, I'll introduce the last speaker. So the final talk is given by Adriana Galván. Adriana is a professor of psychology at UCLA, where she directs the Developmental Neuroscience Lab. Adriana's contributions to cognitive neuroscience have been recognized by our own Young Investigators Award, as well as awards from APS and APA, and recently by the Troland Award from the National Academy of Sciences. The title of Adriana's talk, which hopefully will emerge on the screen soon, is The Promise of Developmental Cognitive Neuroscience.
Awesome. Okay, great. Hi, everyone. I think we're supposed to be done at 3, so I will try my best to run through this as quickly as possible. I'm going to talk about the last 30 years of CNS and how it has informed adolescent brain development. My research has been focused on how changes in the adolescent brain support changes in cognitive behavior. When I started graduate school, it was clear that there was a lot of research on how brain development during the early years support the very important key developmental milestones during early life, such as walking and talking and bonding with caregivers. But in the past 30 years, thanks to CNS, we've learned that adolescent brain development is also necessary to support key developmental outcomes that are necessary for becoming an adolescent and an adult. Surprisingly, we don't yet know what it means to be an adolescent from the perspective of cognitive neuroscience. We know it's a period of transformation from 10 to 25 that is jump-started by puberty, but ends at about the early 20s to 25. And I put up the slide to showcase the various stages of adolescence, including early adolescence, late adolescence, and clearly someone who is between the ages of 11 and 19 are very different. But the brain and the changes that support it are very distinct. And so we've learned a lot about that from tools that we use in CNS. I'll describe some of these things throughout the talk today. And the first is to identify and highlight the fact that cognitive neuroscience has allowed us to identify adolescence as a distinct neurodevelopmental period of life. Many of you have probably seen this image before, and it was something that was published by Jay Gede's work in 2004, showing that the brain develops from the back of the brain to the front of the brain. And one of the reasons this happens is because of changes in gray matter volume or development. This image illustrates some movies. It's a snapshot of movies that were shown a lot, showing that by the time that you are in your early 20s, the gray matter volume that has diminished over the course of development is consequential for how we are able to mature in prefrontal cortical development. So how we're able to think about the future or have goal orientation. And all of that is supported by brain development. We've also learned that there are significant changes in connections among different brain regions. It's not a coincidence that parts of the brain that are responsible for goal orientation and thinking about the future are also more highly connected to emotion regulation and reward sensitivity. And all of this happens during adolescence. When I started graduate school, there was very little known about the adolescent brain, and Nick did a wonderful job showcasing how there's been a lot of advancement in understanding the infant brain and the early childhood brain. And what we've learned about the adolescent brain, which is the next wave of plasticity, is that there are heightened sensitivities in reward-sensitive regions that help us learn about the changing social landscape, and thinking about how risk-taking may be beneficial to becoming an adult. In this study, we had shown, by the way, I should say that when I started graduate school, there was very little known about reward sensitivity in the brain. And that was thought to undergird a lot of understanding about why adolescents make choices that adults deem suboptimal. And so we started by examining how reward sensitivity may influence behavior. In this study, we had shown a group of children, adolescents, and adults pictures of coins and offered them money that they could earn. And what we found is that, similar to what we expected from animal research and adult research, that there was great engagement of the striatal systems, in that adolescents showed an exaggerated response compared to kids and compared to adults in how they responded to reward. And we went on to show this with stimuli that were not simply money, but primary rewards like juice or attractive faces or other things that adolescents found rewarding. We also showed that this was associated with risk-taking behavior. So what we were seeing in the laboratory was also associated with what behaviors adolescents told us that they were engaging with outside of the lab. That was one way to demonstrate that the adolescent brain was much more nuanced than simply thinking about adolescence as a time when puberty struck and had influence over behavior. In 2018, the journal Nature showcased a series of studies highlighting how important adolescence is. And one of the things they showcased is that puberty is really important for producing significant biological, social, and behavioral changes. In this picture that you're seeing here, particularly on the right, you're looking at the height velocity per year as a function of age in girls and boys. And it shows that growth is very rapid in infancy and then slows down a little bit during childhood. But when puberty strikes, the growth rates accelerate at the onset of puberty and then decelerate a little bit at the later stages of puberty. And the reason this is important is because it has consequences for the behavior that we often observe during adolescence. The slide here is showing a review paper from Jen Pfeiffer's group illustrating that different hormones have implications for different regions of the brain. So it's not the case that increases in testosterone or estradiol have the same effects across the brain, but in fact that, for example, testosterone has implications for the medial prefrontal cortex. And if we think about how these brain regions are specific to behaviors, then we can better appreciate how the changes during adolescence as related to hormones may inform a little bit more about why adolescents behave in the ways that they do. These images on the right from Megan Herting's lab show that obviously there are distinctions between males and females, and there are interesting interactions between hormone levels and sex and Tanner staging, which is the way in which we categorize how advanced adolescents are in terms of their pubertal development. Another way that cognitive neuroscience has helped us understand the adolescent brain is in recognizing and honoring the individual differences that characterize this period of life. In this image, what you're looking at is a group of adolescents who are all the same age. They are all in the same grade, but they look vastly different, or I hope that you appreciate how different they are. Does anyone care to shout out how old you think these people are or what grade they're in? 12, 13, what else? 9, okay. They're not 9. They're 8th graders who are all 14 years old. And if you look at this child here kind of in the middle here, this blonde child, he looks like he might be in 4th grade, whereas the kid next to him might be in high school. And this is just to illustrate the vast developmental differences. So when we talk about adolescence, we obviously group people together because that's how we collect our data. But in fact, there are significant differences among them based on their physical attributes, but also in how those physical attributes relate to the social and emotional changes that they are undergoing. And cognitive neuroscience has allowed us to do that, particularly with the advent of big data and the ability to collect data from a vast number of adolescents. In this image from Sarah Jane Blakemore's group, I don't assume that you can see the details, but I show it to illustrate the vast individual differences in structural development that we've collected among key brain regions in adolescence. We know that there are changes in the amygdala, the striatal, the ventral striatum, the prefrontal cortex. And if you were to look at these data more closely, you would see that obviously not all adolescents show the same pattern as their peers, but yet all of them are related to how they interact with the world, how they adapt to the changing social landscape. And importantly, we know that the variability in brain maturation is as great within ages as it is between ages. This image is from Dojenbach and colleagues in 2010, published in Science. And their goal was to identify a brain age. So they used functional connectivity data and mapped it against age to illustrate the fact that although adolescents show the same general pattern with an increase in development as they hit puberty, that there are significant individual differences among them, particularly between boys and girls. And the reason this is important is because we often use age as a way to inform policy that is related to young people. And age is an imprecise index of cognitive maturation and ability. It's the only index we currently use to determine eligibility for most privileges in the United States, including driving and serving in the military or purchasing alcohol or cigarettes. And so it is a bit limiting because we don't have a current way to capture how that variability may inform the policies that we know are important for serving young people and the sanctions and laws that inform their development. We've also, in the past 30 years, recognized that environmental influence has a significant influence in adolescent behavior and outcomes. This image that you're seeing is from Bronfenbrenner, who established the ecological systems theory, which is perhaps the most dominant view in development, showcasing the fact that our environment and our development is not just informed by our own behavior, but by our microsystem, our families, our macrosystem, our society, and our macrosystem, our environment, our government, for example. And what we've learned and what we try very hard to incorporate in our research is that culture cannot be ignored. Culture is defined as a system of values, of practices, of beliefs that constitute our environment, and it's shared by a certain group and transmitted across generations. Eva Telzer and her group at UNC recently did a meta-analysis in which they concluded that there is a greater need to call attention to culture, to the study of brain and development, that we've often included participants in our studies based on convenience, people who are available to participate in our studies, but yet if we don't include the important value of culture, we are missing a big piece of the puzzle. She and her group found that of the 3,700 participants that they studied in this meta-analysis, 3,700 studies that they examined, only 80% did not report the ethnicity of the participants, the socioeconomic status of the participants, and that is a big omission because then we don't have a greater understanding of the environment in which these participants are reared. Particularly because we know that although 25% of the world's population is between the ages of 10 to 24, so people who constitute this period of adolescence, 9/10 live in low and middle income countries, and yet 90% of our current data about adolescence comes from high income countries. So we're missing a big part of the adolescents who comprise our our society and our world. This is important when we think about the growing interest in understanding the relation between family income and brain structure. Kim Noble at Columbia has been a leader in understanding how family income and poverty is related to adolescence and adolescent brain development. She published a study in 2015 that was quite pioneering related to family income and brain structure, in which she showed that for every dollar in increased income, the increase in children's brain surface area was proportionally greater at the lower end of the family income structure. These are the findings from the study. But I want to highlight that for those who have lower income, what her data showed is that every incremental increase had a greater proportional impact on brain development. And all of this is to suggest that we cannot ignore the importance of environment and how brain development occurs. At the microsystem level, that is at the individual level, we know that adolescents' own practices have an impact on how they develop. And in my lab, we've been studying, or spent a lot of time studying the relation between basic needs and brain development, and in particular, sleep. Sleep is a basic need that suffers during adolescence. We know that across ages, how much we sleep or the quality of our sleep has impacts on our behavior and our development, particularly in the brain. But during adolescence, we know that there's a particular relationship between sleep and brain development. This happens because, related to puberty, there are circadian rhythm changes. That is, there are melatonin changes, so the hormone that is released when the sun goes down that induces sleep in all individuals in our species occurs later during the day in adolescence. So for adults, it happens when the sun goes down, but for adolescents, it occurs at 8 or 9 p.m. So that means that biologically, adolescents are not feeling as sleepy as adults do. This is also influenced by bedtimes and wake times, so adolescents go to bed later as they progress through high school, but they also wake up earlier because of school start times. And obviously, there's increased technology use before bed. The reason this matters is because sleep is associated with general health and well-being. So even as adults, if we don't sleep well, we don't have the same emotion regulation. We don't have the same ability to learn about the environment in which we are placed the next day. It's also associated with reckless driving during adolescence, which continues to be the #1 cause of death among U.S. adolescents. It also has implications for policy, and I'll talk about that in a little bit. The brain regions that are most impacted by poor sleep in adults are also those that are undergoing brain development during adolescence. This figure is from Matt Walker's group up at Berkeley. And Matt does research with adults doing sleep deprivation studies in which he deprives adults of sleep for about 48 hours. And what he finds is that they show enhanced engagement of the striatal systems, lower engagement of prefrontal systems. Those are the exact same regions that undergo development during the period of adolescence. In a study from which we are still analyzing data, we examined the sleep environment across adolescents all over Los Angeles, and this allowed us to examine how socioeconomic status may influence the sleep hygiene of young people. They did a sleep log, they graphed actigraphy, which is a sleep watch over 2 weeks, and we also asked them to come to the lab and perform a series of cognitive tasks while they received an fMRI scan. We found that consistent with data from adolescents all over the United States, our participants had averaged about 7 hours of sleep. But what I'm showing you in this figure is that there was a lot of variation or variability in their sleep among those 2 weeks. So some adolescents pretty consistently got 7 hours of sleep. Some adolescents got 5 hours versus 8 hours of sleep. And the reason this is consequential is that in contrast to the belief that only sleep duration matters in terms of behavior for adolescents, sleep variability is also really important. We found that when we looked at brain connectivity in development, that there was significant variability, and that those who received worse sleep also showed dampened connectivity among key brain regions relative to those adolescents who reported better sleep, or who reported better sleep as based on their active graph. And I didn't go into too much detail, but in this study, we got the opportunity to examine the sleep environment. So we examined how well these adolescents slept in terms of the sleep temperature in the room, the light in the room, how many bed partners they had, or how many room partners they had. And what we found is that there was a significant relationship between the sleep environment and their brain development. We assumed that how much technology they had consumed before bed would be associated with their brain development, but that's not what we found. And that's probably because most of the adolescents had a ceiling effect. All of them were engaging in a lot of social media before bed. But what we found instead is that those adolescents who reported lower pillow comfort, or who had suboptimal mattresses before bed, they were the ones who showed suboptimal brain connectivity. And this is just to illustrate the fact that sometimes the most simple environmental factors are the ones that contribute the most to the brain developmental effects that we're interested in. I'll skip this just in the interest of time, but these data suggest that there's a relationship between sleep variability and connectivity among brain regions that we know are important to develop during adolescence. I've written a few reviews about the need for sleep in the adolescent brain and the factors that contribute to it. And just highlight here that during puberty, there's a sleep phase delay. So adolescents are not biologically inclined to sleep at the same time as adults. And that sleep quality is also important for achieving key developmental outcomes. A lot of research on brain development in adolescents and in younger years has been important for linking developmental science to policy. The most consistently relevant ways in which biological science, and in particular neuroscience, has contributed to policy has been in the domain of juvenile justice. This is a snapshot of a recent amicus brief, or at this point not so recent amicus brief, showing or illustrating how a few neuroscientists contributed to understanding the discussions around how and whether adolescents should be sanctioned as adults if they were accused of a crime. And these data, particularly in Miller versus Alabama and Jackson versus Arkansas, were used to argue that it was unconstitutional to hold adolescents accountable, not to hold them accountable, but to sentence them to a life imprisonment without parole if they had committed a particular crime, knowing that cognitive neuroscience has shown that adolescents' brains are not fully developed until the early 20s, well beyond the age of majority in the United States, which is age 18. There are further implications for policy that are more nuanced. These images are from Richard Ross, in which he has examined sleep conditions in adolescents who are in juvenile detention. And as someone who studies sleep in adolescents, the first thing I see are the harsh lights in the right-hand picture, the thin mattresses also in the right-hand picture. And to think about how knowing the importance of sleep in emotion regulation and in learning about the environment, how they may impact adolescents who are in juvenile detention. There have also been implications for school start times. We know that adolescents are biologically inclined to go to sleep later in the night, but yet they wake up as early as younger people. And this is because school start times start pretty early in the United States. In 2014, the American Academy of Pediatrics argued that all adolescents should start school no earlier than 9 a.m. That has not happened in the past 6 or 7 years. And in part, it's because we live in an adult world, and we first consider the needs of adults without recognizing the importance of biologically informed developmental practices. And finally, I'll note that there have been implications for policy regarding how our obsession with adolescent sports, in particular football, may have repercussions for the ramifications of brain development, particularly in terms of sports-related concussions. I'll take a brief note to note that methodological limitations in understanding the brain have been addressed by a few large consortium-level data, including big data. The Human Connectome Project, the ABCD study, the PING study have all done outstanding work to address some of the issues that we've faced when we studied the adolescent brain, because there is a lot of individual difference. There are a lot of considerations in terms of how we can accurately image the developing brain. The large sample sizes have allowed us to examine individual differences, have allowed us to map out functional network topography and large-scale patterns of functional organization. The data sharing among groups across the country has been really beautiful, and seeing that these really senior scientists have been able to coalesce around particular tasks or methodological approaches. It's allowed us to connect structure to function and behavior. Unsurprisingly, one of the greatest challenges in working with young people is that they wiggle. And so we have to contend with the fact that there's a lot more data loss than when you work with adult subjects. And I just want to acknowledge the fabulous work that some of our colleagues have done to address these methodological issues. One of the greatest problems is in head motion in adolescence, and Damien Fair, who has recently moved to Minnesota, or I guess maybe 2 or 3 years ago moved to Minnesota, he and his team have developed tools that allow us to, in real time, examine how much data we're losing, and this has done a lot to save data. So here what you're looking at is a snapshot of the firm software that many of us use in cognitive neuroscience who study adolescents and young people to ensure that we are capturing the most data and saving the most data. There are also limitations of big data in developmental cognitive neuroscience. I know that big data has brought a lot to our understanding of the developing brain, but perhaps this has also led to some limitations, including less nuance in task design. If we're all using the same task, then maybe we're not being as creative in the tasks that we use. It's also challenging to modify protocols that are deemed suboptimal once the data has begun across many different sites. There's greater participant burden or fatigue because they come to our labs and spend hours doing the same tasks. So the last thing I'll say is that cognitive neuroscience has allowed us to shift the cultural narrative around adolescence. A few years ago, the journal Nature published a collection of articles that beautifully synthesized the growing body of research on adolescence through the lens of disciplines that ranged from medicine and social science to education and neuroscience. And the lead article noted that adolescence research must grow up. So instead of pathologizing adolescence and thinking about adolescence as a time of life in which we just need to endure, there are actually really good reasons why the brain develops in a particular way that supports our transition into adulthood. And more recent research has suggested that we are able now with cognitive neuroscience studies to challenge stereotypes of adolescence, to reframe it as a window of opportunity rather as a window of concern or pathology. A few years ago, if you look at the headlines, this is a lot of what you might see, that teenagers' brains are crazy, that they are nuts, that we need to contend with the terrible teens. But we must recognize that if we have that narrative, then we're not supporting young people, that we are not addressing the fact that nature doesn't make a mistake, that it is no coincidence that we have greater reward sensitivity, that we have this protracted development of the prefrontal cortex during a time in life when we are contending with a changing social landscape. It also doesn't recognize that adolescence itself changes across generations. This is a figure from Twenge and Park, published, it was actually 2017, not 2019, in which they tracked characteristic adolescent activities from the mid-1970s through the 2014. In the mid-70s, a lot of adolescents drove a car, tried alcohol, dated. But in 1997, there was a precipitous decline in these characteristic adolescent behaviors. And so I showed this to illustrate the fact that we can't treat adolescents in the same way as we have historically, and that they have unique developmental needs. Cognitive neuroscience tools have helped us shift stereotypes about adolescence. One is in the form of learning, and the second is in pro-social behavior. So I don't have time to go through all of the data, but a few years ago, Daphne Shahami and I, Daphne's at Columbia, and her then graduate student, Julia Devedo, were interested in understanding how the adolescent brain learns, knowing that the striatum is a key learning system, and knowing that the striatum is engaged in an exaggerated way during adolescence. Might that make adolescents better at learning from the environment? And the answer is yes. So I won't go through all these studies. But what we found is that learning systems are uniquely responsive during adolescence and beneficial for learning. This graph is from our study, but others have also shown the same finding, that increased striatal activation in adolescence benefits learning. And so that's just one way that we're beginning to shift the narrative about what is so unique about the adolescent brain and how has cognitive neuroscience helped us understand that. The second is that prosocial behavior increases during adolescence. There's a narrative that maybe adolescents are a little bit selfish or really thinking about themselves as they transition through puberty, but research from brain imaging studies suggests that, in fact, the social brain helps them become more attuned to thinking about other people and how they can support others in their social network. So I'll just close by thinking about what we can do better. We know that our tools are important, but our tools are expensive. So if we want to make sure that they optimize the environment for young people, what are the things that we can better do to support that initiative? One is that we can invest in adolescence and adolescent research. In this 2018 article from that Nature series I mentioned, there was an article that noted that a modern healthcare system without a focus on the unique challenges of pediatrics or geriatrics would be unthinkable. And yet, there is no similar effort on behalf of adolescents. We can use cognitive neuroscience to identify ideal points of intervention. We know that there is great plasticity during the adolescent years, and yet we don't have particular studies that can point to the plasticity that we recognize exists. We can eliminate a disdainful attitude towards young people. And finally, we can pay more attention to adolescents beyond their social media use, which is very pervasive right now in the media. In a recent article, my colleague Andrew Fellini and I noted that young people need experiences that boost their mental health. So beyond focusing on how the pandemic or climate change influences adolescent understanding and the way that their brain develops, we can focus on how learning, how prosocial behavior, how explorative risk-taking can benefit and is necessary for the unique developmental needs that constitute adolescence. So with that, I will thank my lab members, my alumni, my funding, and my collaborators, and all of you for listening. Thank you.
Thank you, Adriana, and thank you, everybody, for sticking around. I hope that you enjoyed this symposium and celebration of CNS at 30 as much as I have. And I know we just scratched the surface of the foundations and frontiers, but we have 4 more days of meeting to go, so there's a lot to learn, and strap in because there'll be another 30 years ahead of us. Take care.